Okay, guys, we just finished talking about epigenetics as one way we control genes, right? Those are things that are going to affect the DNA itself. But now I want to talk about transcriptional controls and how things will affect the eukaryotes' transcriptional factors. So we can do this before transcription occurs, right? And we talked about this when we were talking about the inducible and repressible operons, right? Where we were talking about trip operon and lac operon. Those are things that would be considered pre-transcriptional, and those happen in bacteria. In eukaryotes, is a little more complicated. And this is going to control for the production of those transcription factors to either prevent or activate transcription of the target genes. Now, there's lots of things we've talked about. We've talked about signal transduction. We've talked about how the signal pathways work. And if the transcription factors are there, then the gene will be transcribed. Those signal pathways, as well as epigenetics, both can stop transcription. So those would be pre-transcriptional control mechanisms, right? They happen before transcription. So if you look at this picture, a transcriptional control is where there are things maybe binding to the DNA, like DNA binding proteins that will prevent transcription from happening. But when we get into post-transcriptional, we have the RNA. Now what are we going to do with it, right? Are we going to stop it? Those would be post-transcription. This controls whether or not the mRNA even can leave the nucleus. If we don't process it properly, if we don't add the caps, if we don't add the tails, if we break it down in some sort of way, then it's not going to leave the nucleus. And if it doesn't, it never finds a ribosome and it never does its job. You can add on these RNA binding proteins that will actually stop it. Now you're thinking, why would I make the protein if I don't need it? Well, or the messenger RNA. Well, sometimes we just don't have a control mechanism in place that would stop us from making it, but we can stop it from functioning. Maybe that's a chemical that comes in contact with the cell, and the cell's like, oh, I'm not going to need this, right? The trip operon is one of those things. It's like it's going to make tryptophan as long as it needs it, but once it has enough tryptophan, we stop. Well, maybe that's what's happening in these post-transcriptional. Translational controls, on the other hand, the messenger RNA is in the nucleus. Now what? Okay, so this is where we interfere with the RNA being functional at some point. So the messenger RNA itself can be destroyed prior to translation of the protein. There are some viruses that inject RNA into your cells, and your body has mechanisms to destroy RNA. That's why we have to process our RNA. We have to put those caps and tails. Well, what if something's wrong with the cap or the tail? And those aren't on there right. Either they don't leave the nucleus, or once they get inside the cytoplasm, the cell can destroy them. I want to talk about this. This is something new that has only been around in knowledge for the last few years, like 10, 15 years. It's something called microRNA, miRNA. So what's going to happen here is we have these small messenger RNAs that will be, the microRNAs will bind to the messenger RNA. Sorry, my brain is a little slow. So if you see right here, the miRNA is binding to the messenger RNA. And when it does, it can cause a couple responses. There are genes that are coding in your DNA that are for these small RNA molecules. And they're very specific sequences, right? They're still specific sequences because we're going to bind by base pair recognition. So it doesn't bind to any RNA. It has to bind to very specific ones. And that will be these miRNAs are non-protein generating, right? They're just little micro RNAs. And they can actually physically block the ribosome from attaching to the messenger RNA, so it doesn't physically fit anymore. So that's here on the right. I don't know if you see that down here. So they're going to block translation, and they just physically do it. Another option, though, is that they can actually tag the microRNA in such a way that they will be destroyed. And if you destroy all of that messenger RNA, then you're going to make less of those proteins. Again, this is another control mechanism, right? We don't want to always stop it at the DNA level, or we maybe haven't got the mechanism built in place, but this can work. So how about once we've made the protein? If we've already made the amino acid or the polypeptide, how do we control that? Well, unnecessary proteins just can simply be destroyed. Like if you start building proteins you don't need, your body can break that down. We've talked about fasting in this class a little bit. One of the things that you do when you fast is you actually use up all the worn out cell parts and all the little things that are floating around inside there. So this is an opportunity, like if you go 16, 18, 24 hours without eating, every once in a while, your body can wear down and break up all these little pieces and rebuild them into just plain amino acids that can be used for the next thing. These unnecessary proteins can be simply destroyed. They get tagged by a very small molecule. There's specific names for them. I don't expect you to know them. They get tagged by these small molecules, and that small molecule says, oh, I need to destroy you. And it will actually take it to a protein right here called ubiquitin. And this molecule will actually destroy these tagged molecules. And so if you've got more protein than you need, or if you've got the wrong kinds of proteins or something's detected, we can tag them and be them destroyed, kind of like lysosomes break down worn out parts. This is going to break down worn out proteins. So the last topic I want to talk about, I'm going to talk briefly in our lecture notes, but we're actually going to do 2 labs in class to talk about some of these techniques because I want you to see them in action. So gel electrophoresis is a technique that will allow us to take pieces of DNA and separate them based on the size of the piece of DNA. Now we can cut the DNA at very specific sequences and say, hey, we want pieces cut with this enzyme. And these enzymes have come to us very specifically, usually from a bacteria that has something called bacteriophages. A phage is something that can destroy the bacteria. Bacteria can get sick and die too. So they've evolved the ability to cut up foreign DNA that they don't want in their bodies. And so we've been able to take the enzymes that the bacteria have made and use those to cut DNA as we want. So what we do is then once we've got the pieces cut, we can put them inside a gel. It's like these little, there it is. You can put it inside these little wells right here. I don't know if you can see the little holes right here. We run an electric current through the gel from beginning to end, and the smaller pieces will travel through this porous gel further than the larger pieces. So over time, we can separate it and we'll know exactly what the bands are. Now, scientists use this for organismal DNA fingerprints, right? So we can take an organism, cut it with a very specific sequence of enzymes, and then we'll see how far the specific bases are. Now, we do this with humans. We do it for crime scene evidence, right? It's a little more complicated than, they show us on CSI, but basically there are techniques used to chop the DNA the way we want, to amplify it so we have enough of the DNA to run the gel. And then we run it. And if we treat everything exactly the same, then we're going to see the same prints for things that are identical to each other, things that are identical. We can do it for familial identification. So if we don't know who the father is, or if you want to find out who's your uncle or whatever, those kind of things. And then, of course, evolutionary trees. We've done it a lot with COVID, right? The COVID virus has a nucleotide sequence inside of it, and we've been able to chop it, cut it, and actually identify the difference between alpha variant, delta variant, Omicron variant, and so on. And we do that by biologically sequencing. Now, you probably have heard PCR if you've had a COVID test. There's 2 different kinds of COVID tests. There's a rapid test that looks for a specific protein that the virus makes, and then there's the PCR one, which is actually looking at their DNA. The PCR test takes a little longer, has to be sent away because they have to test it in a different way. And what they'll do is they'll take the DNA and RNA of a molecule and they can cut it up, pull out the piece they're looking for, and make millions and billions of copies of that specific piece of DNA. And then they run the gel, right? Then they run the gel. So what you'll do is you'll take the sample of the DNA, you'll put it together with a bunch of different chemicals, a specific polymerase, RNA primers, nucleotides, etc. You put it in this tube and you run it through the process of DNA replication over and over and over. So these small segments of synthetic DNA tell us which specific sequences we're looking for, right? It's for a specific sequence. Then we chop that guy out and then we copy. It rapidly replicates the DNA. And then, of course, we use it in the gel. We don't want to put one piece of DNA in the gel. That doesn't do any good. We need millions and billions of copies. So we put PCR and gel electrophoresis together. So the labs we're going to do in class, they're going to have done the PCR portion for us. They're going to have processed the RNA pieces for us. So we'll have RNA that's already been processed to run through our gel. Another type of technology that's being used with DNA is genetic engineering. Because that genetic code is universal, remember the chart, right? The AUG and all of that. Scientists can transform bacteria or insert genes into other organisms. We're going to do a lab where we're going to actually insert some genes. We're going to knock out other genes, and we're going to specifically look at what happens by knocking out a specific gene, one that we're looking for. But one of the things we do now is we can actually insert these genes into a plant or into an animal, and we can actually make it do specific things. If you remember, plasmids are those little circular genes. We can trick a cell to take in the plasmids. You heat it and you cool it and you do different things to it, and it'll cause it to take in the plasmid. RNA transcription and translation, they're going to happen to whatever DNA is inside. They don't care. So if you take in a gene, like from a virus or something, your body is going to make those proteins. And so what happens here is this specifically is one where they're inserting a gene called the BT gene. It kills a certain kind of boll weevil kind of thing that eats the plants. And so they insert this gene into the plants genetically. We grow the cells into whole plants, and those whole plants now have that inserted gene. That inserted gene could have potentially some side effects, which is why some people are a little freaked out by GMOs. We use it a lot for agriculture. We're growing crops that are disease resistant, famine resistant, drought resistant. We grow ones that'll increase the crop yield. We grow ones that the insecticides that not the insecticide, the herbivores, the herbicides that are put on them. Like if you've heard of Roundup, Roundup is an herbicide. It kills weeds, but if you spray it directly on your crops, it'll kill them too, unless you've genetically engineered that crop to handle Roundup, and so on. So we're doing all this kind of genetic engineering, inserting genes. We're using it to create medicines, right? Insulin, for instance, and some different things. So by being able to take a gene from one organism and put it in another, we can do this. You've probably seen some of the weird things that we've done with it, like make glowfish. Like if you go to the store, you see these tanks that have like UV lights and the fish are like glowing neon colors. That's genetic engineering that's done that. I think it's kind of ridiculous, but that's just my personal opinion. Like, why do we need glow in the dark fish? But they've inserted like the firefly glow gene, the one that makes the chemical that makes fireflies light up. They've inserted it into cats. If you look it up, it's really crazy. The glow in the dark cats. We're using it to make biofuels, right? To make biofuels so we can get off of fossil fuels. So there's a lot of different things we're doing. So many more that I'm talking about. The last technique I want to mention, it's not an AP tested question, but I want you to understand it because it's really recent. And one of the labs we're doing is on CRISPR. And so CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Blah! It's a big fancy word for looking at very specific sequences that are palindromes. They're the same forward and backwards. We pronounce it CRISPR, and Cas9 is the specific enzyme we use. And this technique is using those bacterial's natural immune systems to cut DNA at specific locations. So we're splicing genes. Now, some of these genes we're cutting, if we can cut a gene that's bad, we can prevent it from being expressed, right? And so that's called knocking out a gene, right? We knock it out. And so we're now working on editing genes, right? We can have a precise pair of scissors, because DNA is really hard, small, precise pair of genetic scissors that can go in and cut out those bad genes and target very specific sequences. So Cas9 is the little enzyme that's responsible for doing the cutting. And so if we know what sequence we're looking for, we'll learn more about this in our lab. If we know what sequence we're looking for, we can actually cut that specific gene. Scientists are researching this on ways to deal with things like high cholesterol, HIV, Huntington's disease. What's really crazy is they've actually recently been able to knock out the sickle cell anemia gene in an individual and cure them. What they've done is sickle cell anemia makes hemoglobin that's defective. If you destroy that gene, the body will use its fetal hemoglobin gene. So hemoglobin is needed. So you have two types. You have fetal hemoglobin, which is used when you're in the fetus. And then when you become an adult, you start using regular hemoglobin gene. If we kill the regular hemoglobin gene, then your body will revert to using your fetal hemoglobin, which won't have the mutation. So the person that's been cured of sickle cell anemia has no longer shown any sickle cell traits from this. And it's fascinating to me because this has happened in the last 5 years. And we're learning so much more about how to cut these genes and how to knock out these genes. So if we can find ways to knock out specific cancer genes that are causing tumors where they shouldn't be, and so on and so on and so forth, it's really fascinating. So we're going to do a lab, like I said, where you're going to get to do some CRISPR and knock out genes and see that it actually works. And you don't visually see us cutting anything. It's just kind of a lab technique. But I wanted to mention it because when you go into college, you're going to run into CRISPR if you study science at all. All right, guys, I'm done. Yay! Last unit, last lecture.